Hi, everybody. Welcome to today's GlycoNet webinar. My name is Warren Wakarchuk. I'm the Associate Scientific Director of GlycoNet. The first speaker we have is Professor Lynne Howell, who is at the Hospital for Sick Children in Toronto. She's also a professor in the Department of Biochemistry at the University of Toronto. Lynne got her PhD in the UK at the University of London and did postdocs at MIT and at the Institut Pasteur in Paris. And she joined the Hospital for Sick Children some time ago. And she was cross-appointed to the university shortly thereafter. Lynne's work is in structural biology, and she's been doing some really fascinating work that she's going to tell us about today on polysaccharides that are involved in biofilm formation. She's currently a Tier 1 CRC chair in structural biology, as I said, at the Hospital for Sick Children. And without any further ado, Lynne, I will pass it over to you.
So I'd like to start by thanking the organizers for inviting me to the GlycoNet webinar series and this virtual opportunity to speak to a broad section of our community across the country. I hope everybody is safe and that research plans are now well underway for everyone. I know my lab was very excited to be able to send 3 people back to the lab this week. The current situation and this outbreak has caused me to modify in part my presentation, mainly because typically I start my presentation with a question and a show of hands. It's kind of difficult when you're talking to a computer to necessarily see those hands. But I'm going to assume for a minute that the audience I have today is much the same as the audience that I would regularly get. And that typically, when most of you think about bacteria, you think about bacteria in the planktonic state as free-swimming planktonic cells, single-cell organisms. If you don't take anything else away from my presentation today, I would like you to at least realize that for the most part, bacteria live in what's called a biofilm state. And what a biofilm is an aggregation of cells enclosed in a self-protected matrix, which is represented in the blue here, and frequently attached to some kind of surface. The reason why most of the time that bacteria live in this state is because this state, or the biofilm, provides a number of different advantages over the planktonic growth. It provides localized gradients and provides habitat diversity. It provides the opportunity for the bacteria to absorb chemicals and nutrients. It also helps to retain enzymes extracellularly that are capable of digesting food for the bacteria. It provides both synergistic, cooperative, and competitive environment to both live productively with other bacteria, but also as an environment to compete against other bacteria. And from the point of view of this talk today, and from a health perspective, the bacteria are also provided some tolerance and resistance to antibiotics in the immune system by living embedded within this matrix. As a consequence, it's not hard then to assume or understand that biofilms have become a ubiquitous problem in the healthcare setting. They cause both device-related infections and tissue-related infections, a host of which are represented here. In fact, over 80% of all chronic infections are biofilm-related, and estimates of the annual cost to the American healthcare system certainly is in the $20 billion range per year. From a perspective of the work that we do, we are interested in a number of these, such as bacterial biofilms on endotracheal tubes, chronic presence of bacteria in chronic wounds, and also in the lungs of cystic fibrosis and other lung infections. I'm not actually going to talk to you about the translational work we do today, because I have a couple of stories that were recently published that I'd like to share with you. Let's take a little look more at what a biofilm is. This is a micrograph here of, in fact, B. cereus, a gram-positive organism that grows sort of in a filamentous type way. And what's not clear until I artificially color the matrix is that the matrix really does form the glue that holds this collection of cells together. The matrix, this red material here, can contain a number of different components, such as proteinaceous adhesions, extracellular DNA, and I wouldn't be here today if it didn't also include exopolysaccharides. The exopolysaccharide component of the biofilm has a number of different roles in biofilm formation. It's involved in the adhesion of the bacteria, here in red, to the surface. It also is involved in helping with cell-to-cell interactions. And as alluded to previously, it's also important from the role of protecting the embedded bacteria from both antimicrobials as well as the host immune response. Bacteria are actually incredibly adaptable and have typically the genetic capacity to synthesize more than one exopolysaccharide. This is obvious on this plate here where we've grown colonies of my favorite model organism, Pseudomonas aeruginosa, that were isolated from the sputum of cystic fibrosis patient with chronic lung infections. What you can see on the plate is sort of two different types of colonies. One here represented in the sort of diffuse mucoid-looking colony. And this is typical of the production of a polysaccharide called alginate. The smaller little colonies are not an artifact of this and not a contamination. It's actually a second type of Pseudomonas aeruginosa colony called rugose small colony variants, or RSCVs. And these are associated with the production of two different types of polysaccharides, both PSL and Pel. And it's the work that we've done on the Pel polysaccharide that I want to tell you about today. The gene locus responsible for the production of the Pel polymer was actually first identified by a transposon mutant library performed by Frieder and Coltman back in 2004. What you can tell from these little diagonal triangles in the top of this gene cluster is that these represent each an individual transposon insertion, whereby if you disrupt any one of the genes with this transposon, they found that you no longer got a biofilm at the air-liquid interface. Our project on Pel began in about 2009, and as things so often do, it began as a conversation over a glass of wine at a meeting, in this case a FACID meeting in Arizona, with Matt Parsek, who is at the University of Washington, who has become a long-time and very productive collaborator. What Matt and his group had recently done was they had developed a method to actually be able to turn on the production of this 7-gene operon using arabinose. And when you enable that and add arabinose to the bacterial culture, you end up with this very thick biofilm at the air-liquid interface. If you grow these bacteria on a Congo red plate, you get this wonderful wrinkly colony morphology. And both of these are characteristic of the production of the Pel polymer. What Matt had done to expedite our structured functional study, he's also created deletion mutants in each and every single one of these genes, but any one of them has the same effect in that if you delete one of them, you get no pellicule, so you get a cloudy, turbid culture, no biofilm at the air-liquid interface, and you get a smooth colony morphology when you're looking at Congo red. Again, indicative of no Pel production. Using these strains, we were able to do a number of studies that have established, first and foremost, what the chemical composition of the polymer actually is. So using gas chromatography mass spectrometry, GCMS, analysis of these strains, we were able to show that the polymer is composed, in this particular strain, predominantly of N-acetylgalactosamine and some N-acetylglucosamine in approximately a 5-to-1 ratio. We've shown in other studies that these two sugars can actually be deacetylated. And as a consequence, this deacetylation will provide a cationic polymer characteristic. By the characterization of one of the enzymes involved in the and present on the operon, we now have data that suggests that the polymer linkage is an alpha-1,4 linkage, although we have yet to actually demonstrate this using NMR analysis of the actual polymer. But the enzymatic activity that we've demonstrated certainly is alpha-1,4 linked. So we think that PEL is a random polymer of predominantly N-acetylglucosamine that is linked in an alpha-1,4 linkage. What Matt and his colleagues have been able to do is to show that PEL is incredibly responsible for the structural integrity of the biofilm. It localizes to the stalk of the biofilm. So what I've got here is a flow cell growing a biofilm, which you can see in this mushroom shaped, and it's labeled with, the biofilm biomass is labeled with cytose 62. And the polymer is labeled with a lectin, in this case WFL lectin or Wisteria fluorabundin lectin, that is specific for GalNAc. And what you can see as you just look at the signal for the lectin, you can see that the polymer associates with the base and the surface where it attaches to the cover slip. In other data that I won't show you, but I will summarize, is that the cationic nature also helps PEL to interact with eDNA, which also co-localizes to the bottom of the biofilm. And it also, again, given its cationic nature, helps protect against minor glycoside antibiotics, which are by themselves positively charged. So we know that it's involved in structural stability, and it interacts with DNA. And my role and the role of my lab in this project has really been to think about the following question. It's like, how does these 7 genes synthesize the polymer? How is the polymer modified, and how is it exported from the cell? And this is really the question, the overarching question that has been driving the research in my lab for the last 12 years. Like any good bioinformatician and structural biologist, we use a series of bioinformatics tools to sort of take our linear set of genes and generate a 3-dimensional image or model of what the components might do. We incorporate, things about the localization, whether or not it's lipid anchored, whether or not it's in the outer membrane, periplasmic, etc. What I want to do today is actually talk to you about the inner membrane components and the work of Gregory Whitfield, who recently graduated from the lab with his PhD, and 2 stories that have developed from his thesis. If you look at these 4 proteins, we have in these 4 proteins a protein called Pelf that is involved potentially in the polymerization. It looks like a glycosyltransferase. We have a protein PelD, which looks like it might be involved in signaling or the regulation of the production of the polymer. PelE has predicted in parts of its, certainly in the cytoplasmic parts of its protein structure, to be involved in potentially in the scaffolding or protein interactions. And we're still not quite sure exactly what PelG does, although it contains similarities to mate transporters. So maybe it's involved in the transport of the actual polymer. However, given that PEL is predicted, PelF, which is the polymerase, is actually predicted to be in the cytoplasm, how is polymerization and export into the periplasm actually coupled? So the first question that Greg asked himself and asked and answered is, does PelD, E, F, and G actually associate to form an inner membrane synthase complex? This has some precedence in the field in that both cellulose and alginate, as well as PNAG, all have a synthase complex that are involved in the production of the sugar. So I'm going to show you just one piece of data at this point in time, which will support the the hypothesis that they really do form a complex. And this is an experiment where we have co-immunoprecipitated each of the proteins in turn in a background in which we have deleted one of the proteins, one of the genes, in this case PelD. We've then complemented the strain back with either PelD or PelD with a tag. And we've used that tag to then pull down not only the protein with the tag, but any other protein that will come with it. So you can see in this, when we tag PelD, we can pull down PelD very nicely. We have specific antibodies for each of the 3 proteins listed here on the screen. So we can show that we've pulled down PelD, and with it we can pull down both E and F. If you look at the reciprocal experiments, where in this case we've tagged PelE or PelF, we can see similarly we can pull down each of the other ones. We verified these experiments using mass spectrometry, so we're fairly convinced that in each case, each of these proteins somehow associates and can be pulled down in a co-immunoprecipitation experiment. So we can begin to sort of build a network of interactions. But co-immunoprecipitation doesn't actually tell you whether or not these interactions are direct or indirect. And we had a problem in that we could not get any information about PelG because it's in our hands. It's been unstable when detergent solubilized. So to begin to actually then sort of put the pieces together, Greg turned to a number of other different techniques. And I'm just going to summarize the results here in an overview rather than going through the actual results. First of all, he used the technique of bacterial 2 hybrid, whereby you can tag pairs of proteins and look for direct interactions between the components. He also actually did an additional trick where you can take the bacterial 2 hybrid and actually add a 3rd protein. So you can actually look at what happens and how the proteins interact in the presence of not just 2, but also 3 proteins. And doing that, as you can see from the black lines, he was able to establish interactions between almost every single one of the proteins that are present. He also looked at the localization, specifically of PelF, because it was predicted to be periplasmic, and obviously it doesn't, as we alluded to, if it's not associated potentially with the rest of the proteins in the inner membrane, PelD, E, and G, then how is it going to actually be coupled to export into the periplasm? We saw a codependency of PelF's localization on each of the 3 proteins, but predominantly with PelG. And last but not least, we were also able to look at the stability of the inner membrane proteins, in that if you delete one of the proteins and then look at the level of proteins in the other 2, what we found was that the levels of the other 2 were compromised when you start to delete one of the others. Again, providing circumstantial evidence that in fact all 4 of these proteins associate to form an inner membrane complex. So while we are hoping and working towards purifying the whole complex, we have, for the minute, taken a divide and conquer approach to our structural studies. And the first protein that we actually started to look at, because we thought it was originally cytoplasmic, although now we know it's associated with the inner membrane, was actually PelF. We actually haven't been able to crystallize PelF to date, but we were able to use it to show that it could bind UDP nucleotides using isothermal titration calorimetry. We can see a binding isotherm here for UDP, but not for TDP or GDP, suggesting that obviously that you need a UDP GalNAc sugar nucleotide for the production of the polymer. What was interesting though was that we could not detect any binding of the UDP GalNAc and therefore no transferase activities in vitro, which sort of suggested to us perhaps that it needs to be part of that complex, and that maybe there are some kind of conformational changes that are occurring in the transferase that need to occur for it to be activated to bind the sugar nucleotide. So this then started making us thinking about how is Pel synthesized, or at least how Pel synthesis is regulated, and how does this regulate does this regulation affect the complex? Because you can imagine that you want to be able to turn off and turn on the polymer biosynthesis. In elegant studies done in 2007, Vincent Lee, when he was a postdoc in Harvard, had demonstrated that the second messenger, cyclic di-GMP, was absolutely required for PEL biosynthesis, and that it interacted with a protein called PelD. We've solved the structure of PelD and shown that it contains a so-called GAF domain and the degenerate GGDF domain, and that these two domains at their interface bind dimeric cyclic di-GMP. As a consequence of knowing the structure, we were able to map out residues that are involved in binding. And we can show that that this isolated cytoplasmic domain not only binds the cyclic di-GMP, again shown here through isothermal titration calorimetry, but we also contains a robust biofilm. But then when we knock out and mutate arginine 367 to alanine, we find that we not only abrogate the binding of the nucleotide, but we also no longer have biofilm formation. So we use this to actually start to interrogate whether or not there was a change in the complex if you no longer were able to bind cyclic di-GMP. Because there was precedence in the field that for PNAG that suggests that you actually need cyclic di-GMP for a stable formation of complex. So the first experiment that we did was actually looked at the cellular localization of F using sucrose gradient fractionation. We have a cytoplasmic component and an inner membrane component, and we used NADH oxidase to show that we had separated these effectively. NADH oxidase is only present in the inner membrane. What we found, as you can see here, was that we actually have an equilibrium between the cytoplasm and the inner membrane. When we look at this in the background of a PelD that contains this mutation that no longer binds cyclic di-GMP, we found that again there is a equilibrium between the two, albeit that if you actually densitize and quantify this, there is a slightly less PelD in the inner membrane than there is in the wild type protein. The second experiment we did was to look at this mutual stability. And in the parental or wild type strain, as I've shown you previously, what happens if you delete PelD, what I said was that you had alterations in the stability of other proteins. So you can see in the absence of PelD, PelE is no longer very stable, and we can't see it very well in our Western blot. However, we can complement it and recover activity and recover the signal for the protein, which also appears to be present when we actually have this abrogated binding mutant of PelD. Again, if you quantify this, we do see a significant decrease in the amount of PelD that's present, suggesting that while the complex does form, perhaps it is improved, and the stability of this complex is improved when cyclic di-GMP is bound. There's one other clue to potentially get an idea of how we think this might be working and a model for what might be happening, and that is our structure of the PelD cytoplasmic domain in the presence and absence of the nucleotide indicated that there was a small but significant rotation of the GAF domain relative to the GGDF domain, suggesting that there might be some kind of conformational change that might occur in this protein that might be transmitted to the rest of the complex when cyclic di-GMP bound. So our model over here on the left is the following, and that is that when cyclic di-GMP binds to PelD, it undergoes a conformational change that is transmitted to Pelf, and that this conformation now allows the binding of UDP catalysis to occur and the subsequently polymerization to occur. And we think that, that PelG is primarily the portal the portal through which the polymer actually emerges. So I've shown you the structure of PelD. We also now have additional support for the protein-protein interactions. We've solved recently a structure of PelE, which has again similarity to proteins involved in protein-protein interactions, so-called TPR and 1433 proteins. We've also spent a lot of work and time looking at the outer membrane, and I'm going to briefly summarize those here. In this case, we have a protein PelC, which polymerizes into a dodecamer. We think it forms a funnel, an electrostatic funnel that attracts the positively charged polymer to the bottom of this electronegative surface and helps with the export of the polymer. We've also characterized the TPR domain of PelB. We've shown that it interacts with the protein PelA. And we've also characterized PelA and shown that it's an active glycosyl hydrolase. This is a point in time when I would normally start to tell you about our translational work exploiting the glycosyl hydrolases for therapeutic purposes. But today I actually want to talk a little more about another exciting development that we've had. And that stems from the fact that we're getting a bigger picture of the polymers and the proteins involved in this production of this polymer from a perspective of a gram-negative organism. And when we started, this was a fairly niche polymer. It was involved only in the production of Pseudomonas and other pseudomonads. But we realized as you increase the number of bacterial genomes that are sequenced, we have now come to realize that in fact that it's actually prevalent and there is over 465 species of mainly of Proteobacteria that actually have the genetic capacity to actually produce the PEL polymer, making the results that we have in our structure function studies much more relevant to a host of potential additional pathogens. Whether or not these pathogens obviously or bacteria produce the polymer is yet to be determined, but they certainly contain the genetic capacity to do so. However, we realized that there was an inherent flaw in our analysis and that we'd used the protein PelC, the outer membrane protein PelC for this analysis, and therefore we were biasing our results to only to finding other gram-negative organisms. So in a parallel study with my colleague John Parkinson at SickKids, we started to look for if we actually took every single one of the PEL genes from Pseudomonas and actually started to use those to look for other genes across the entire fully sequenced genomes, whether or not we'd find any additional polymers, or at least organisms that were able to produce the PEL polymer. And what, much to our surprise, we found initially was that the gram-positive organism Bacillus cereus appeared to have two proteins that were similar to PelG and PelF. A closer look at the genomic context of those genes revealed 3 additional genes that similarly had homology to PelD, E, and A, as well as a couple of additional auxiliary genes that I will tell you about in a minute. We've now expanded that analysis and found that in fact greater than 165 different gram-positive species appear to have and contain what looks like a pel-like loci. What Greg Witt has gone on to do was to determine whether or not, at least in the model organism B. cereus, whether or not this pel-like loci really does produce a pel-like polymer. So the first thing that he did was obviously establish that the organism itself actually produced a biofilm. Here is the biofilm at the air-liquid interface, and we can label it and color it with crystal violet. And we get a very robust signal. He's deleted each and every single one of these genes, and in each and every single one of our cases, when you does that, we lose this biofilm biomass and we have no signal. But that doesn't tell us whether or not it's PEL. We can also see in a micrograph, we can see that the wild type has a lot of matting in between all of the B Cereus cells, but there's absolutely no matting or existence of a matrix of whatsoever in the PelF deletion. But none of these experiments tell us whether or not this is PelF. So we turned to the use of our lectin that we'd shown previously had been specific for PEL. And we looked at both wild type and the PelF deletion mutants and saw that there was a signal for PEL in the using the lectin in the cell associated. And we also had now access to a PEL antibody, and again, the wild type strain, but not the deletion mutant, gave us a signal, suggesting for the first time that the B Cereus material probably has some chemical similarity to the polymer produced by Pseudomonas. We were able to exploit our hydrolases to look to see whether or not the polymer was the same. As I said previously, we have characterized the PelA hydrolase to be an alpha-1,4-N-acetyl galactosaminidase. Using this enzyme and preformed B Cereus biofilms, we can actually degrade the biofilms with fairly good IC50s in the nanomolar range in the wild type, but not a active site mutant. Again, providing evidence to suggest that this polymer is the same, has the same chemical structure. One of the auxiliary genes downstream of the operon is actually another potential hydrolase. And so we expressed and purified this hydrolase from B Cereus. And similarly to the experiment I just showed you, we can take this enzyme and we can also degrade the B Cereus preformed biofilms using a wild type, but not a mutant. And the final test was to take this particular enzyme from B Cereus and look at the Pseudomonas aeruginosa biofilms. And again, the hydrolase is able to degrade the biofilm, but not the mutant, all providing circumstantial evidence that the polymer produced is actually the same. Production of this polymer is also looks like it is controlled by cyclic di-GMP. There is a PelD ortholog. We can model the structure of the B Cereus based upon the Pseudomonas structure we've done. We can identify residues in the active site that are important for binding the nucleotide. And we can use, again, isothermal titration calorimetry to show that the wild type protein can bind the dinucleotide, but that any one of these residues that are involved in binding the nucleotide abrogate its binding, and similarly abrogate the formation of biofilms. So we're fairly convinced that this polymer also is regulated in a comparable manner through cyclic di-GMP. Similarly, Greg went one step further in this particular case, and there had been a recent identification of two enzymes, one involved in the production of dicyclic di-GMP and the other involved in its degradation. And we could again go back to our dot plots and our lectin and our antibody and look to see what happened when we actually deleted the enzymes that make the cyclic di-GMP, where low levels of cyclic di-GMP would be present. In this particular case, if you haven't got the polymeric, this nucleotide, and you can't bind it to PelD, we don't get any signal, we don't get any polymer. And when we have, we mutate and delete the enzyme that's responsible for degrading the cyclic di-GMP, we're going to get elevated levels of that nucleotide and elevated levels in our either dot plots of the WF lectin or the antibody, to the point where actually we are now beginning to actually see polymer in the secreted fraction. So what I hope I've been able to show you today is that, excitingly for the first time, we've identified PEL machinery in gram-positives. We've been able to show and identify proteins that are involved in the production of the cyclic di-GMP. We've shown that it is similar to Pseudomonas aeruginosa. The polymerization seems to be regulated by cyclic di-GMP. And there are a lot of similarities that we can draw from the two systems that we have now established. Excitingly, and during the course of the current shutdown, we've taken this project one step further forward, and we're looking at annotating a strep intermedius strain that similarly uses Pel polymer for at least certain strains of strep intermedius produce a Pel-dependent biofilm. And we've used the time productively during our lockdown to actually annotate a transposon mutant biofilm experiment that we've done to show and to begin to get some additional clues as to how in this particular species biofilm formation might be regulated and other components that might be important for this process. This is particularly interesting to us because strep intermedius does not actually have the capacity to actually produce cyclic di-GMP, and therefore the regulation has to be, of the polymer production, has to be different in this particular case. And with that, I'd like to thank all of the team assembled here in one of what will become, I'm sure, a ubiquitous set of photographs on Zoom for lab meetings and for the foreseeable future. Each and every one of them has been involved in the annotation project, and we're going to dedicate the resulting paper to the COVID-19 lockdown. In addition to the current members of the lab, I'd like to, point out, I think I've pointed out Greg Whitfield, who was responsible for a lot of the work that I've presented today, but he was also assisted in this by Lindsay Marmot, and also in the past, Greg Whitfield and others. This is also a collaboration with a large number of people. The work I've told you about today involved John Parkinson in the bioinformatics, Joe Harrison, Matt Parsek for teaching us a lot about how to manipulate bacteria, as well as Don Shepard and his postdoc Francois Lamoff, who have been critical in helping us characterize some of the enzymatic activities of the hydrolases. With that, I'd like to also acknowledge the funding sources that have provided support to the projects over the years. And with that, I would like to end and ask, open this up to any questions that you might have. Thank you.